Okay, in this assignment today, I'm going to go over how to make a mitosis storyboard. Now, if you recall from our discussion in mitosis, the whole goal is to start out with a parent cell and make two daughter cells that have the same number of chromosomes. These are cells that are going to be identical to the parent cell. So for human beings, we would start out with 46 chromosomes and end up with two new cells that also have 46 chromosomes. For my example today, so I don't have to draw 46 chromosomes, I'm going to start out with a diploid cell of 4 and end up making two new cells that also have 4 chromosomes. And this process is what is used for our what we call somatic cells. And remember I said that somatic cells in your body are just regular body cells, such as bone, skin, muscle, blood. Those are all considered somatic cells. Now, if you recall, in order to get this whole process started, before we actually can do mitosis, we have to get a cell ready to actually divide. And in order to do that, you have to take a smaller cell that was just created and give it an opportunity to get bigger. So it's a period of growth. We're going to call this period of growth interphase. And from the lecture, you'll also remember that in an interphase, this is also a period of time when the chromosomes unravel into a state of DNA, also known as chromatin, and then make copies of themselves. So I'm going to use blue to demonstrate the chromosome that was given to this organism by its father. There's the number 1 chromosome, and I'm going to make a smaller one to represent number 2. That means that mother also had to give this organism another 1 chromosome and a smaller number 2. And of course, when you make copies of them, they end up being attached at the centromere to one another, like so. So I now have a total, even though this organism's diploid number is 4, now, if you count them up, there's actually 8 chromosomes here. And that's because when this cell divides, I'm going to have to send 4 to each of the new cells. So now that the interphase is over with, we're ready to go on to the next step, which we're going to call prophase. And a couple, 2 or 3 important things happen in prophase. The most significant thing that happens in prophase is that this unwound DNA, which used to be chromosomes, now tightens back up into chromosomes. And not only do those chromosomes tighten back up, but they also make their way over. They start to take the journey to get towards the center of the cell. So I'm going to go ahead and draw the original and the copies of these chromosomes and draw them as if they are making their way towards the center of the cell. And you'll also notice that the nucleus, which I didn't draw over here, I need to draw it, there was a nucleus here. You'll notice the nucleus is pretty much dissolved and gone. So after prophase, we're now going to move into a phase that's called metaphase. And quite simply, the most important thing that happens in metaphase is these chromosomes that I just drew randomly align themselves down the middle of the cell. And I'm drawing both of Dad's here in order next to each other. That doesn't necessarily have to be the case at all. The other significant thing that happens, besides those moving towards the center of the cell, is from a little structure called the centriole, the formation of what we call spindle fibers starts to occur. And we're going to find out in the next phase, which is called anaphase, we're going to find out what actually these spindle fibers, what their purpose actually is. So in anaphase, the centromeres actually split, and what is going to happen is a copy, the original and the copy, are both going to start to make their way over to the opposite sides of the cell. And the belief is that the centromeres actually break down the spindle fibers, and that is how they actually make their journey to get to the sides of the cell. And that's anaphase. Last, and you can see that the only obvious thing left to happen is we need this cell to pinch off, and this pinching off is what we call telophase. So in telophase, I'm going to show that in each of these new cells, we now have 4 chromosomes, which is our diploid number that we were after in the first place. And we're also going to see the reappearance of our nucleus. So these are going to be brand new little baby cells that are going to be ready to actually become big cells and undergo this whole process again. Plant cells, if you recall, they don't have the ability to pinch, and so if we were to do this for plant cells, we would actually draw the exact same thing, but instead of the pinching off, you would see a cell plate built down the middle, and it would just kind of break off. So again, this is the process of mitosis, and this creates clones. It's also known as asexual reproduction, and it creates identical cells with the same number of chromosomes as the parent cell.